Hello and very welcome. Hello and welcome to our very important lecture about G protein receptors versus enzyme linked receptors. G protein systems, G inhibitory proteins, G stimulatory protein receptors versus enzyme linked receptors. Okay, so before we talk about the differences, we have to establish the similarities. The best way to always learn about the differences between two different categories is to always begin with the similarities. The similarities between enzyme linked receptors, enzyme linked receptors, and G protein receptors. Similarities are both of them are forms of second messenger systems. So here we're no longer dealing with first messenger systems. We're no longer dealing with steroid hormones. We're no longer dealing with a steroid hormone that is able to translocate from the intravascular compartment and then go through the interstitial fluid and then cross the lipid bilayer and then cross the cytoplasm and then cross the nuclear envelope and then swim through the nucleoplasm and then ultimately bind the DNA and transcribe for us our target protein. That was steroid hormones. That was lipophilic hormones. That was non-polar hormones. We're no longer dealing with that. Now we're dealing with hydrophobic hormone, with lipophobic hormones, with hydrophilic hormones, with non-steroid hormones. These non-steroid hormones are now an example of second messenger system because obviously due to their structure being non-polar, due to their structure being polar and hydrophilic and lipophobic, they're unable to cross this lipid bilayer. So what they do is they bind to an outside. So in both cases, whether we're dealing with the G protein receptor, the ligand, the stimulus, the hormone, the first messenger has to bind on the outside. In enzyme linked receptor, the signal has to bind on the outside. Now what happens intracellularly in the intracellular signal transduction pathway that happens inside the cell, that is what makes the G protein receptors different from the enzyme linked receptors. So now let us begin with the very simple enzyme linked receptor. In enzyme linked receptors, what we have is, obviously we have an extracellular portion, and this extracellular portion is going to bind our stimulus, is going to bind our first messenger, which is always non-steroid. Because if it was a steroid hormone, it does not need a second messenger. It can just cross the lipid membrane. But what it does now, it binds to an outside portion. And upon the binding of the first messenger, the non-steroid hormone, to the outside portion of the receptor, what happens is there will always be a intercellular, there is always going to be a conformational change. That conformational change is going to activate for me an enzyme that is linked to the receptor. So now the receptor is attached to an enzyme. Like here, the JAK enzyme, the Janus kinase enzyme. This Janus kinase enzyme, for example, one of the biggest examples of this enzyme linked receptor is the leptin hormone. The leptin hormone is very important for the regulation of appetite and energy conservation. When leptin binds to the extracellular receptor, what happens is a conformational change will take place that will eventually culminate in the activation of the enzyme that is linked to the receptor. So the enzyme linked here is the Janus kinase. So a kinase is obviously an enzyme that phosphorylates. It promotes phosphorylation. That's why they call it a kinase. So the Janus kinase Once it's activated by this conformational change of the receptor it is bound to, it can either do one of two things. That's why I have the number 1 and the number 2. So the first thing is that it can either this phosphorylated Janus kinase. So the Janus kinase was only phosphorylated because of the conformational change. And the conformational change in the receptor only happened because of the hormone was able to bind to the signal, to the receptor. If the hormone did not bind the receptor is going to be in the off, inactive position. And when the receptor is in the off and inactive position, the Janus kinase is going to be inactive. It's not going to be phosphorylated. So the P is not going to be attached to the Janus kinase. This conformation is only when the hormone binds to the receptor. When the hormone binds to the receptor, a conformational change will happen, and that will lead to the Janus kinase being activated. And when it's activated, it gets phosphorylated. And this phosphorylated Janus kinase can do one of two things. The first thing is that it can either phosphorylate other additional enzymes, and these other additional enzymes will give us our target response. So that's probability 1. Phosphorylation of the Janus kinase was based on the conformational change of the receptor, and the receptor conformational change was based on the binding of the hormone, and that led to the phosphorylation of the other target enzymes that gave us target response. In probability 2, what happens is the phosphorylation, the binding of the receptor to the hormone is going to make some conformational change in the receptor. And the conformational change in the receptor is going to activate for me the Janus kinase. And when the Janus kinase is activated, that means it's phosphorylated. So now the Janus kinase that is phosphorylated is now going to phosphorylate for us a very important special enzyme known as STAT. STAT means signal transducer and activator of transcription. So now you know where this is leading to. When the the phosphorylated Janus kinase phosphorylated for us the STAT is now going to translocate through the cytoplasm into the nuclear envelope and then bathe and swim through the nucleoplasm until it binds a location in the DNA, and that's going to give us transcription, and then we're going to transcribe a target mRNA, and this target mRNA is going to be translated in the ribosome, and that will give us our target protein that will give us the cellular effect. In essence, the enzyme linked receptor is a form of a second messenger where you have you don't have a G protein that is linked to the receptor. You have an actual enzyme that is attached to the receptor, and that enzyme that is linked to the receptor is inactive, as well as the receptor is always inactive except when the ligand binds to the outside membrane. So when the ligand binds to the extracellular membrane, that causes a conformational change, and that conformational change is going to ultimately activate for us the linked enzyme. That's why they call it enzyme linked receptor. The linked enzyme is going to be activated by being phosphorylated, meaning adding a phosphate group, and then a phosphate group was added, and that could give us one of two ultimate outputs. The first output is going to be the phosphorylation of other target enzymes, and these phosphorylated target enzymes will eventually lead to a target response within the cell. The other probability is for the Janus kinase, the phosphorylated Janus kinase, to phosphorylate for us STAT, signal transducer and activator of transcription, and that is now going to migrate to the inside of the cell, and that after it migrates to the inside of the cell is going to bind a particular promoter region in the DNA to transcribe for us the DNA. after transcription happens, we get a target protein, and that target protein is an enzyme that will give us the target effect. So either way, we have to first have the ligand, the hormone, the first messenger bind on the outside, then the conformational mechanical change will lead to the phosphorylation of the linked enzyme, in this case Janus kinase in the leptin, and then that will ultimately lead to one of two things, either phosphorylating cytoplasmic proteins or migrating, translocating mobile receptor into the nucleoplasm, and then binding the DNA to transcribe for us target mRNA that will be translated in the ribosome to give us target proteins. That is a leptin receptor. That is the leptin receptor. Now let us talk about the other forms of second messengers, and that is an example of G protein. So now what happens in the G protein is that before we talk about G protein coupled receptors, let's quickly get out of the way the concept of calcium calmodulin. So another type of second messenger system is that calcium is always a lot on the outside of the cell. Calcium is found a lot on the outside of the cell. Calcium has the kinetic energy to diffuse down concentration gradient from an area of a higher calcium concentration on the outside to an area of a lower calcium concentration on the inside. But can it do that? It cannot. Why? Because the channels are closed, the calcium channels are closed, so it cannot cross and enter. So it will deflect, it will remain sequestered on the outside. But when first messengers bind to the target response, to the target extracellular receptor, that's gonna lead to the opening of the calcium channels, and then calcium will flow down two concentration gradient. Down chemical concentration gradient by diffusion, so now we're moving chemically, mass effect, from an area of a higher calcium concentration to an area of a lower calcium concentration. That is a chemical gradient. What is the other gradient? What is the other gradient? That is going to be electrical gradient. So outside there is more electropositive, and inside there is less electropositive, more electronegative. So there is another favor for calcium to come in. So now calcium is going to obey two gradients: chemical gradient to diffuse inwardly, and electrical gradient to diffuse electrically. And then calcium will enter the cytoplasm, and once calcium has entered the cytoplasm, it will bind calmodulin. Calmodulin is an inactive enzyme in the absence of calcium. But when calcium enters the cell, it will bind to calmodulin, and then calcium calmodulin together will make a complex known as the calcium calmodulin receptor. And then this calcium calmodulin complex is either going to activate very important enzymes in the cytoplasm or translocate into the nucleus and bind the DNA, leading to transcription of target enzymes in the target cell. So that is as far as calcium calmodulin. We got that out of the way. Where is my eraser? We have gotten that out of the way. Now let us now go back and talk about the G. By the way, calcium's effects can either be by binding to calmodulin, but in other instances, the flow of calcium is not the target response when it binds to calmodulin. It can either bind calmodulin, or calcium itself is going to cause like smooth muscle contraction or affect the secretion of the cell. But we will talk about that some other time. All you need to know for now is that the binding of the first messenger outside can sometimes lead to the opening of the calcium channels and the influx of calcium down electrochemical gradient to cause the target effect. Now we have gotten everything out of the way, we have left the best for last, and that is the G proteins. Let us now talk about the G proteins. The G proteins now, two systems. We can have a receptor that is attached to a G protein. This G protein can be bound to GDP, and it could also be bound to three subunits: the alpha subunit, the beta subunit, and the gamma subunit. So how many attachments do we have? We have the receptor, that's component number 1. We have the G protein, that's component number 2. We have the GDP, that's component number 3. We have alpha, that's component number 4. We have beta, that's component number 5. And we have gamma, that's component number 6. These are 6 components. This is the configuration of an inactive G protein. So this right here, where you have a G protein receptor attached to a G protein. So that's the receptor, that's the G protein attached to it. That's why they call it G protein coupled. So because they are coupled together. The G protein receptor coupled to a G protein, coupled to GDP, coupled to alpha, beta, gamma. This is the off position. Now there is no receptor bound to the hormone. That's the off position. That's the minus position. That's the inactive position. We are not working now. There's no job. We are off now. When the first messenger binds to the G-coupled protein receptor, now we have to change. We have to change from the off into the on position, from the minus into the plus position, from the inactive into the active position. So how does that happen? What happens is, this is very important. What happens is the signal, the hormone binds to the receptor, and then when the hormone binds to the receptor, what happens is GDP is going to be hydrolyzed into GTP. So it used to be guanosine nucleotide that is attached to 2 phosphates, PI1 and PI2. Guanosine, that's why they call it guanosine diphosphate, because it only has 2. Once there is activation, now we get more. So now it's no longer going to be guanosine diphosphate, it's going to be guanosine triphosphate, PI1, PI2, and PI3. So now it's rich because we are now activated, we are now on, and we are now active. Now it's guanosine triphosphate. So now what happens next is that this guanosine triphosphate, this GTP, is going to recruit the alpha subunit. So now we will have an extra complex known as GTP alpha complex, the GTP alpha complex. So now when we are activated, we have a GTP alpha complex, and we have the G protein coupled receptor coupled to the G protein, and now there is no GDP, because now we have converted to GTP. So now we have two enemies. Now we have GTP alpha as a complex. The other complex is our receptor and the G protein, and the gamma and the beta sitting all sad, missing that their alpha is gone and their GDP is gone. So now we used to have 6, now we only have 1, 2, 3, 4. The GDP is no longer there. The GDP, so now we are in the on position. We only have our receptor, the G protein, the beta, and the gamma. The GDP has been converted into GTP, and the GTP took alpha with them. So now this complex GTP alpha is going to go and it's going to activate for us adenyl cyclase. And adenyl cyclase is going to convert ATP. ATP means adenine triphosphate. So now it's an adenine nucleotide with 1 phosphate, 2 phosphates, 3 phosphates. is going to convert it into adenine monophosphate. So that means it's subtracted 2. We subtracted 1, we subtract 2, and we became adenine monophosphate, AMP. We went from ATP into AMP. This AMP is going to activate for us protein kinases, turn protein kinases from the off position into the on position, from an inactive position into an active position, from a minus position to a plus position. These are kinases. And remember, all kinases add phosphate group. So when a kinase is activated, the kinase is going to phosphorylate enzymes. So now the kinase is going to phosphorylate for us our target enzymes. So the target enzymes are now going to be turned from off position to on position, from inactive into active, from minus into plus, and then they will do our target response. This does not go on forever. The target response, when we have achieved our target response and we have achieved our homeostasis, what happens now, the GTP, the alpha is going to say to the GTP, goodbye, I miss my old friends. So alpha is now going to hydrolyze GTP back to GDP. And then we will have alpha GDP complex that's going to go back, and then GDP is going to bind the G protein, and the alpha is going to go back to the old friends, and we will have GDP, G protein, alpha, beta, gamma attached to the G protein receptor that is now off, because now we have finished. But once the stimulus binds, all of these changes happen. G protein will hydrolyze GDP to GTP, then GTP is going to recruit alpha, and then GTP alpha complex is going to activate adenyl cyclase. Adenyl cyclase is going to convert ATP into AMP. AMP is then going to activate for us protein kinases. Protein kinases are going to phosphorylate target enzymes, and then target enzymes are going to give us target responses. So this is GDP, G-coupled protein receptor. By the way, this adenyl cyclase, some other G-coupled protein receptors, they use guanylyl cyclase. So the same pathway, but the only difference is that, for example, the epinephrine and the norepinephrine, they do not utilize adenyl cyclase, they utilize guanylyl cyclase. So now we're no longer going to be dealing with ATP being converted to AMP, so we're not going to be dealing with adenine group, we're going to be dealing with guanosine or guanine group. Okay, this guanine group is going to be changed. Okay, there's one more thing that I forgot to mention. So this cascade, this is the appropriate cascade if we are talking about activation. So the binding of the receptor leading to the activation of this intercellular G protein activity. That leads in the end to the activation of target enzymes. So if that happens, then the G protein is known as G stimulatory protein. So this G stimulatory protein, known as GS protein, is different from GI. I'm going to talk about the difference. So let's begin with GS. GS is what I just mentioned, that when there is a binding of the hormone to the receptor, that's going to cause conformational change. The conformational change is going to activate the G protein, the G stimulatory protein attached to the receptor. The G stimulatory protein is going to cause GDP, meaning guanosine attached to 2 phosphates, to be converted into GTP, guanosine attached to 3 phosphates, Then the GTP is going to recruit the alpha subunit and devoid it. Is going to subtract and remove the alpha subunit and leave behind the beta and the gamma. And you will have GTP bound to alpha. GTP bound to alpha is going to activate adenylyl cyclase, and in other enzymes going to be guanylyl cyclase, but it's the same thing. And if it's adenylyl cyclase, we're going to convert ATP, adenine triphosphate. We're going to convert it into adenine diphosphate, ADP, into adenine monophosphate. But if we're dealing with guanosine, we're going to turn GTP into guanosine monophosphate. But in the end, the guanosine monophosphate or the adenine monophosphate, the only difference here is the nucleotide. In this complex, we have adenine attached to a monophosphate. Here we have guanine. So how many subunits we have? We have adenine, we have guanine, we have cytosine, and we have thymine. So here, adenine monophosphate, and here guanine monophosphate, depends on the preference of the enzyme. And that leads to activating protein kinases, and the protein kinases are going to give us a phosphorylation of our target enzymes, and these phosphorylated target enzymes are going to give us our cellular effects. That's the G stimulatory protein, because the G stimulatory protein led in the end to the stimulation of these target responses. Sometimes this same exact cascade leads to an inhibition of the target response. So now let's say I want to inhibit the target response. What happens is the same thing. The hormone is going to bind to the G protein coupled receptor. The G protein coupled receptor is now going to activate not a GS, G stimulatory protein, but a GI, G inhibitory protein. And the G inhibitory protein is going to do the same thing. It's going to hydrolyze GDP into GTP, and then GTP is going to recruit the alpha, and the alpha is going to either activate adenylyl cyclase or guanylyl cyclase, and that eventually leads to guanosine triphosphate being converted into guanosine monophosphate, or adenine triphosphate being converted into adenine monophosphate. And then the protein kinase is going to be activated, and protein kinase is going to phosphorylate the enzyme. But the target response here, the phosphorylation of the enzyme in the end, is not going to activate for us a target response. It's going to inhibit for us the target response. So now we're going to be stopping and shutting down and inhibiting. That's why they call it G inhibitory protein. The same cascade, but the terminal effect is either going to be stimulatory if we're dealing with a G stimulatory protein, or it's going to be inhibitory if we're dealing with a GI protein. So that's as far as G protein coupled receptor. There are other G proteins that are not going to, whenever they are activated, they're not going to lead to GDP being hydrolyzed into GTP and alpha subunit. No, it's not going. That's not what's going to happen. But there are still G proteins. So the G protein that is attached to the receptor can either go down the line of, activating adenylyl cyclase, but that's not all of them. The other possibility is that when the ligand binds to the G protein receptor, to the G protein coupled receptor, this G protein now is going to go in the cell membrane and is going to activate an enzyme there known as phospholipase C. Phospholipase C. This phospholipase C is going to chop and bring a fragment from the cell membrane, this fragment here known as PIP2. It's sitting in the membrane. This PIP2 is actually known as phosphatidyl. Whenever you hear phosphatidyl, we're talking about cell membranes. Phosphatidyl inositol 4,5 biphosphate, known in shortcut as PIP2, phosphatidyl inositol phosphate 2, PIP2. What phospholipase C does is that it will take PIP2 and cleave it into two products, product number 1 and product number 2. Product number 1 is known as IP3, shortcut for inositol triphosphate. And this inositol triphosphate is going to have very important cellular effects, leading to an increase in calcium, intracellular calcium concentration is going to increase. So where did this intercellular calcium come from? Did it come from outside, down concentration gradient? No, we're no longer dealing with calcium calmodulin. What happens is the cell has reserves, it has pools, it has reserves of calcium. If you ask anybody about the mitochondria, they will tell you it's the powerhouse of the cell. But they don't know that instead, in addition to being the powerhouse of the cell, it is also the cell reserve of calcium. So it will start to squeeze calcium out of the mitochondria, and there will be a lot of calcium in the cytoplasm. Not only is calcium stored in the mitochondria, calcium is also stored in the endoplasmic reticulum. So inositol triphosphate IP3 is going to squeeze, in addition to squeezing the mitochondria, is also going to squeeze the endoplasmic reticulum, leading to intracellular calcium concentration increase. And when this calcium concentration increases inside the cell, that leads ultimately to target effects such as smooth muscle contraction, and affecting the secretion of the cells. So that's one target response. So now we have achieved our target response. The other effect is the activation of DAG, diacylglycerol. So this diacylglycerol now is going to go and activate and phosphorylate for us very important enzyme known as protein kinase. Whenever you hear kinase, you know that this is an enzyme that adds a phosphate groups plus pi. So if a protein kinase finds adenine diphosphate, you know that it's going to make it adenine triphosphate. It's going to add a phosphate. When it adds a phosphate, it's going to convert from an off position to an on position. It's going to convert from an inactive into an active form. And then we're going to have protein kinase activated. And when protein kinase is activated, it's going to give us our cell target response. So that shows you here, we're not dealing with alpha leaving beta and gamma like what happened before, but we're still dealing with G protein. But the G protein is not dealing with alpha and beta and gamma and GDP. It's dealing with phospholipase C. So the G protein is going to activate phospholipase C. Phospholipase C is going to grab a chunk from the cell membrane PIP2, phosphatidyl inositol 4,5 biphosphate. It has 2-phosphate biphosphate at the 4th position and the 5th position, but you can just call it PIP2, phosphatidyl inositol biphosphate. Is going to cleave it into two products, one known as IP3, inositol triphosphate. Inositol triphosphate is going to increase intracellular calcium concentration, and diacylglycerol is going to activate protein kinase C, You see phospholipase C, then leading to protein kinase C, giving us our target response. This is very, very important. The main example here with the JAK and STAT, with the JAK and STAT activation. That's an example of the a main example of enzyme linked receptor is our leptin receptor, or leptin hormone. This hormone that's very important for appetite regulation as well as energy conservation. Yes, I have talked about everything. If you have questions, leave them in the comment section and I will try to solve them. Thank you very much for your attention, and take care.